So we are going to start unit 1 now. First unit, first topic, introduction to pH, pH scale, then pH indicators and pH meter. So what is pH? pH is simple. It is a unit. It's not it's a number actually. So pH is a unit of measure which describes the degree of acidity or alkalinity. That is basic condition. Acidic condition or basic condition. Acidity or alkalinity of the value from 0 to 14. So the formal definition of pH is that. What is the definition for pH? pH is nothing but the negative logarithm of the hydrogen ion concentration. Or in the formula you can write pH is equal to minus log H plus ion within bracket, that is called the molar concentration of hydrogen ion. Then pH value. What is pH value? So the pH value of a substance is directly related to the ratio of hydrogen ion, that is H plus ion, and OH minus ion, hydroxyl ion concentrations. If the concentration of H plus, that is proton, H plus ion is higher than the concentration of OH minus ion, then that solution is acidic in condition because H plus ion concentration is very high. Suppose if the OH minus ion concentration, hydroxyl ion concentration is higher than proton, that is H plus ion concentration, then that solution is called a basic solution. Suppose if a solution is neutral and H plus ion and OH minus ion concentration will be neutralized. So 7 is the neutral pH. 7 is the neutral pH. Less than 7 is acidic condition. Greater than 7 is basic condition. Then coming to pH scale. pH scale corresponds to the concentration of H plus ion concentration, that is hydrogen ion concentration. Based on that only the pH scale is designed. So if you take the exponent of H3O plus ion concentration and remove the negative sign, then you have the pH of the solution. For example, take pure water, H plus ion concentration. That will be converted into pH value. So the pH of that solution would be 7. Because it is having 1 into 10 power minus 7 molar, that is H plus ion concentration. So this is the pH scale. The figure of the pH scale here. So that this is 7, pH 7 neutral. It is neutral. When you go beyond neutral, it's an acidic condition. When you go below the neutral, this is basic condition. So you have color variation, color change when you change the pH. So this is a pH scale, and it is given in pH indicator papers, pH paper. pH paper you can see this pH scale with variation in color. Now pH indicators, strips. This is a pH strip. Red litmus paper. In that you are going to keep a 1 drop of base, then the red litmus becomes blue color. Similarly, this is blue litmus paper, blue in color. You are keeping 1 drop of acid here, then it becomes red. So here the pH scale is given. See that this is actually litmus test using litmus paper. Here pH values of different samples are given. See that water neutral, then milk, tomato juice, apple juice, banana juice, lemon, then hydrochloric acid, highly acidic, most acidic. So the color will change. So litmus paper is used, 2 types of litmus papers, red litmus and blue litmus. So litmus dye is a natural pH indicator. So a litmus paper is a small paper. Here you can see that it's a paper or a strip of paper that has a dyes in it. There is one dye is coated on it. When it touches a base or a dye, when the litmus paper changes its colors depending on the nature of that type. So either it is a base or it is an acid. Based on that, color will change. If a litmus paper is exposed to an acidic solution or acid, it either changes its color from pink, it changes its color from red to pink. If a strip of litmus paper touches a base solution, then the paper changes to a shade of blue color. See that this red litmus becomes blue color, blue litmus becomes red color. Now we are coming to pH meter. Next one, very important instrument. So introduction to pH meter. So a pH meter is an electronic instrument. This is used for measuring the pH of any solution. Whether it is acidic solution or acidity is there, alkalinity is there, it is used for measuring the pH of any liquid through special probes. They are sometimes used to measure the pH of semi-solid substances also. So a typical pH meter consists of special measuring probes. What is there in a pH meter? The measuring probes are, one is a glass electrode and a reference electrode. Two electrodes are. They are connected to an electronic meter that is going to measure and display the pH reading on the display. There is a variation in the concentration. Then this is the reference electrode and glass electrode. Both are combined, and in this beaker solution is the pH. That is unknown sample is kept out there. pH for this unknown sample will be displayed on this display. So this is a pH meter. So a sample which is placed in a cup or in a beaker or placed in a glass probe at the end of the retractable arm, it is placed in it. So the probe is connected to the main box, the main electrical box. There are two electrodes inside that probe that measures the voltage variation in the solution. So one is contained in liquid with fixed pH. The other one measures the acidity of the sample through the amount of hydrogen ion concentration. So a voltmeter in the probe, it measures the difference between the voltages of the two electrodes. So the meter, the voltmeter, then translates the voltage difference into pH value and it displays it on the screen, that is on the display screen. So before taking a pH measurement, the meter must be calibrated. It must be calibrated using a solution of known pH. For a known pH, it has to be calibrated. And next one is a temperature compensation meter. So temperature compensation is contained within the instrument, that is in the pH meter, because pH electrodes are highly temperature sensitive. So you have to maintain the temperature. So temperature compensation only corrects for the change in the output of the electrode, not for the change in the actual solution. So buffers are the solutions we are going to see next. Buffer solutions, they are the solutions that have constant pH values and they have the ability to resist changes in pH. So they are used to calibrate P is equal to power, H is equal to hydrogen ion, H plus in the molar bracket, hydrogen ion concentration. So the pH of a solution can be measured by the pH meter. So the glass electrode is in half cell. It is half cell glass electrode. And the column electron is another half cell. So they both has to compare. So following reactions takes place in the electrodes. The glass electrode, what is going to happen? The glass electrode contains silver chloride and hydrochloric acid. All these remain in the ionized state. Silver becomes Ag plus and electron minus. AgCl becomes AgCl plus Cl minus. HCl, H plus Cl minus. So all the above three, that is Ag plus, H plus, Cl minus. All the three, the equilibrium reactions are balanced. So in the glass electrode, H plus ion concentration is generated. So the thermal electrode contains mercury and Hg, Hg2Cl2 and KCl. Here also the following series of ionization takes place in this thermal electrode. Hg, Hg2Cl2, KCl. Then Hg is ionized, H plus G plus to electron minus. Similarly, 2Hg plus 2Cl minus. potassium chloride becomes K plus and Cl minus. So the above equilibrium reactions are balanced. So in the calomel electrode, H plus ion concentration is not generated. Then the probe, the types of probes which is present in that pH meter, you have a glass electrode and a reference electrode, then third one combination electrode. So first figure is highly sensitive proton. This platinum ion. This is actually a glass. There is a glass tube. Then this is pH meter, and this pH meter, see that it is having platinum ion. Then here you are having potassium chloride solution here at the center, and mercury is here, calamite is here. Then KCl crystals are deposited there. Porous regions are there. Your refill can be done opening this. Then it goes to pH meter. This is the combined cell, combination cell. That is both the calomel electrode and glass electrodes are combined. So wires to pH meter, it goes there. Two electrodes are inside that. This is gel, which is a silver chloride reference electrode. And reference electrode internal solution is here. It's L11. This is the junction. Then silver chloride covered silver wire is here. Another electrode. Glass electrode, internal solution enters inside this. Calibration of the pH. So in pH meter, calibration should be performed with at least two standard buffer solutions that span the range of pH values to be measured. So for general purposes, buffers are at pH 4 and pH 10, both are acceptable for this kind of calibration. So the pH meter has one control, that is calibrated to set the meter reading equal to the value of the first standard buffer, and a second control, that is slope one, which is used to adjust the meter reading to the value of the second buffer. So a third control, it allows the temperatures to be set. Construction of the pH meter. So a pH meter, which is having a power pack, there is power pack this side, electrodes are probes that side, and there is inside the power pack you have on-off switch indicating meter, temperature compensation knob, calibration knob, wire with plug in. And in electrodes or probes you have glass electrode, calomel electrode, or combined electrode, combination of these two, glass and calomel. The measurement of pH, how to measure pH using a pH meter. The zero of the dial is set mechanically. First you have to set 0 in the monitor. So the knob for temperature compensation, there is a knob for temperature compensation. So that knob for temperature compensation is fixed for temperature of the solution. Then the electrodes are dipped into a standard buffer of known pH. Then adjustment it can be done so that the dial reads the value of the buffer solution. Then the electrodes are removed and washed well with the distilled water. Then the electrodes are dipped into the same solution, sample solution. So the dial shows the pH value of the sample solution. So types of pH meter. So this is a manual pH meter. Manual pH meter, digital pH meter. Two types are there. This is manual one, a manual pH meter. You have electrode stand here, set 0 is done here manually, set buffer is here, temperature compensation knob selector is here, then selector is here, the electrode is here, then indicating meter is here. This indicating meter is monitor. And next one is a digital pH meter. The values are coming here digital pH meter. Disorders in human blood and human body. In agriculture, pH meter is used. Then drawing it is used. Then brewing in fermentation industries, pH meter is used. Corrosion prevention, that pH meter is used. Dyeing industries, jam and jelly manufacturing, printing in pharmaceutical industries. All these places pH meter is used. There is main application.